Okay, finally we are moving on to 1.5b, which is DNA profiling. This is where the water gets a bit muddy, so we need to tread the water slowly. The learning intentions, I'm gonna skip over them because we're going to be learning them anyway. So DNA profiling and electrophoresis. We can use DNA to identify people. How do we do that? Everyone's DNA is different. Uh, the DNA contains different patterns of repeated sequences, such as AATT. They could be any pattern. And we can cut up DNA using, not sourcing, using special enzymes called restriction enzymes. The restriction enzyme cuts DNA in every place there is a specific sequence. All right. Now, since everyone's DNA is different, when you use the same restriction enzyme to cut uh, two people's DNA, the cut up segments are going to look different. And we're going to see how some of these DNA technology, such as gel electrophoresis technology, uses this interesting feature to make a DNA profile. Gel electrophoresis is a technique used to separate DNA fragments or other macromolecules based on size and charge. Right. Describe gel electrophoresis. First thing, you get the DNA sample, and second thing is cutting up using the restriction enzyme. Restriction enzyme cuts the DNA at specific places, and then you place the DNA, the cut up segments, in a gel electrophoresis well. You turn the power on and run for 30 minutes. And then you soak the thing in a fluorescent dye over gel. And the last thing you do is shine UV light and take a picture. The picture taken is called the DNA profile. Now back to what is a DNA profile? Well, it's a DNA pattern of bands corresponding to different sized DNA fragments. They are created by for humans, also called a DNA fingerprint because the pattern is unique for each person. This can be used in forensics, genetic screening, and paternity tests, etc. This is an example of the use of DNA profiling in determining the paternity of a child. Now, here you see five people: the mother, the child, the dad one, dad two, and dad three, and their corresponding DNA profile. Now, this is where the water gets a bit muddy, you see. The child's DNA profile has 1, 2, 3, 4, 5, 6, 6 strands. Out of these 6 strands, about 3 strands um, will occur in the mother, and about 3 other strands will occur in the father's DNA profile. So we just look at each strand and match up with the mother. So mother, child, here's one, and there's the second one, there's the third one. And then we look at that 2 and that 3, and we try to do the same thing and that 1. Okay, so that 1 has a strand where the child doesn't have one. This doesn't happen because the child's DNA profile comes from both the dad and the mother. So whatever the child has, the dad must also have. So that 1 is not the real father. So we look at that 2. There's the strand, okay, and there is no strand here because that's mother strand. All right, let's keep going down. There is no strand down here. So notice how these 2 strands, the mother doesn't have it, but that 2 doesn't have one of them. So we look down to keep going to that 3. Okay, where the child has one, the dad also has one. Where the child has 2, but the mother doesn't have, uh, the dad has both of them. So if you um, compare the child, the dad like this, you will find the potential dad of the child. And this is very precise. This is very accurate in deciding the paternity of a child. And this is a legit method used to decide, who's the real father. Why do different people have different DNA profiles? Well, our genome, which is all of our genes in our body. Well, genome means all the genes in a organism. The genome includes two things, the introns and exons. Now, we know what exons are. Exons are the genes that are transcribed, that are later translated, and they are made into protein. Exons are the genes that are expressed in our, physical features, in our uh, features you can see. So, say I have blue eyes. That would be an example of exons being expressed. But I could also have genes for blonde hair, but I have black hair instead. That means the part of gene that have the blonde hair information is an intron because it is not expressed. What are introns? Patterns of introns vary person to person. The base sequence of introns can vary as well, and they have tandem repeat sections. Introns repeat. Right? There are the short tandem repeat and the variable number tandem repeat. One's shorter and one's longer, and they both repeat for sometimes shorter, like 10 times, sometimes even 50, 60, 100 times. It's quite amazing. And here we get into the two types of tandem repeats. As I said, the variable number tandem repeats. These are repetitive sequence that is about 10 to 60 base pairs. This means each unit here is about 10 to 60 units long. That is pretty long considering that it also repeats for a fair amount of times. You can have a unique number and length of these, and they are found in introns. Variations are alleles, and you have a unique combination. And then we get to the short tandem repeats. These are repetitive sequences that are about 2 to 6 pairs long. So each STR repeating unit would be about 2 to 6 bases long. So an STR repeat unit could be AT. But a VNTR repeat unit could be much longer, like ATCG, ATBT, AATT, GGCG, such as DNA profiles. The results of electrophoresis can be used to construct DNA profiles. They may be displayed in an electropherogram or in a table of data. Okay, okay. All right. Electrophoresis. Okay. Now, we have learned about this thing, the result of gel electrophoresis. This is what you get at end of gel electrophoresis. You have bands of cut up DNA segments. And this is using the VNTRs. But with these ones, with these DNA profiles, we're using the STRs. So how this works is there are DNA markers, which is a specific sequence that can occur at specific loci in the genome. And there are sections of DNA that are commonly inherited and can be traced back. They are repeating patterns in the DNA that are found at known locations in the DNA. So there are known locations in our DNA that scientists know that a specific pattern must occur there, that a specific tandem unit, repeating unit must occur there. And you can have variations of each STR noted by different numbers on the table, and you have two copies of each. Now, these dot points really don't explain the whole thing really well, so I'm gonna try to do that myself. So as you see here, STR-DNA marker. Each of these numbers and characters, they all stand for, okay, where was I? Each of these sites. Each of these markers are a specific site in the DNA. And why is this so important? Because scientists know that as at these sites that your DNA intron repeat for a certain amount of time. So for different people, there will be a certain pattern, the same pattern, but repeating for different amount of times. And we keep moving. Mother, 28, 30. What does that mean? Well, that means at this site, at D21S11, the mother in one of her chromosomes have 28 repeats, and in her other chromosome has 30 repeats. Because humans have two chromosomes, they come in a pair. That's how it works. And the alleged father at the same site will have 29 chromosomes in one of his my bad 29 repeats in one of his chromosomes, and in his other chromosome will have 31 repeats. Now, because mother and the alleged father both give one of their chromosomes to the child will have 28 and 31. 28 from the mother, 31 from the alleged father. Now, by looking at this table, we can see that the child shares at least one number of repeat from both the mother and the alleged father. So this way we can confirm that the alleged father is the daddy. Now if I keep moving on, is that the last slide? No. Sequencing of DNA. Now this is something different. Before we were learning about DNA profile, we were profiling of DNA. But that's not sequencing of DNA. Sequencing of DNA means literally figuring out all the base sequences, base pairs of a DNA. Well, what does it mean to sequence DNA? It means to determine the order of base pairings for a gene or genome. Why do we do it? For forensics, diagnose disease, and other purposes. Now, this is much harder, much, much, much harder than DNA profiling. Because what you're dealing with is not just bands appearing on a piece of paper or a table. You're dealing with long sequences of ATCG, ATCG, ATCGs. You are mapping out essentially the blueprint to building an organism, and that is pretty hard. There are a few methods to doing this. There is using the chain terminating nucleotides. So you have the normal nucleotides available when copy DNA. There's ATCG. And chain terminating nucleotides uses DDNTPs. They all have fluorescent marker attached to them. When DNA or Taq polymerase adds one, no more nucleotides can be added. Electrophoresis and DNA sequencing. So, electrophoresis. Okay. Right. Okay. Okay. So the chain terminating methods isn't really explained quite well, but basically, you know what? Let's skip this for now. Electrophoresis. You put the DNA samples in 4 test tubes with all PCR requirements, as well as chain terminating nucleotides. You will have normal nucleotides plus one of the 4 chain terminating ones. What will happen? Well, the contents of each tube is placed in 4 separate wells on a gel. The gel is turned on and run. Fragments in each lane separate. Smallest tube go further. That's not good. I gotta speed up. Analysis of gel bands reveal the sequence of the target DNA. So you have the normal nucleotides plus the chain terminating ones. And given enough tries, because we do this many times, that all of the chain terminating ones have attached to at least one of the um, bases. So after that have been done, we then take all the strands and run it through the gel. So the fragments in each line will separate. The shortest one will go the furthest distance, and the longest, also the heaviest one, will go the least distance. We learned that in DNA profile. Next, we move on to electropherogram. 1.5.5 The results of electrophoresis may be displayed in an electropherogram. 1.5.7 The results of electrophoresis can be used to construct DNA profiles. They may be displayed in an electropherogram or in a table of data. Okay. Electrophoresis. Right. Pillary electrophoresis. That's not important. Okay. Electropherogram. Remember, there are two types of electropherograms. There is this, which is used for DNA sequencing, and you can tell each peak in an electropherogram represents one of the 4 DNA nucleotides. The height of each peak represents the amount of light absorbed and emitted by the fluorescent molecule attached to the DDNTP at the ends of a DNA fragment. Now, where did this come about? Well, it came from the machine that we were talking about earlier. Because of the fluorescent marker attached to the DNA, the machine can detect those fluorescent markers and make a table, literally, a graph, a gram, electropherogram, literally make an electropherogram appear on the computer screen. So it would look like a messy lot of lines in different colors, but they're just your basic DNA base sequence, the primary structure. Gene and genome sequencing. 1.5.6 DNA sequencing enables mapping of species genomes. That's a bit of repeating there. Of species genomes. 1.5.8 DNA profiling identifies the unique genetic makeup. And I see some typos there. Genetic makeup of individuals. Now, I'll just read this. For lots of different organisms, humans, mice, and even a few exotic creatures like seahorses, we have figured out the whole genome. We know on which chromosome specific genes are located, and we also know the order of millions of bases that make up each chromosome. We organize this information into a map of each genome. A genome contains all of the instructions for building and operating an organism, essentially a blueprint. Having a genome map helps us to do things like diagnose and treat diseases, improving crops, track down bases of the inherited traits, and much more. And that's the last slide for you. There you go, 1.5b finished. For 1.6, we'll be moving on to gene tech biotech, which is a bit related to this, but more complicated. So study biology well, so we can be more prepared for upcoming units. They're hard, man. I'll tell you that. They're hard.